We have one final level of protein structure to consider, quaternary structure. We will see that not all proteins exhibit this level of structure. This level of structure pertains only to those proteins that contain more than one polypeptide chain. Each of the chains is a subunit, and we refer to such proteins as oligomers, containing more than one subunit. They may be comprised of multiple copies of the same subunit, as in the case of collagen, which is a homotrimer. Instead, they may contain different polypeptide chains, such as hemoglobin, which is a heterotetramer. It is perhaps important to point out here that in each case the protein is the functional unit. This means that polypeptide and protein are not necessarily interchangeable terms, at least not in oligomers. Let's consider what kinds of forces hold the subunits together. Well, they will be those types of non-covalent forces we've considered thus far: electrostatic attractions, hydrogen bonds, and hydrophobic interactions. There may also be covalent bonds connecting the subunits in the form of disulfide bonds. An interesting phenomenon often results in multi-subunit proteins known as allostery. The prefix allos means other, and it is an appropriate way to denote those proteins in which a conformational change in one subunit induces a change in another, and often adjacent, subunit. The best way to understand this topic is to consider an example, and hemoglobin is an excellent choice for this study. As shown here, it has two alpha chains, pictured in blue, and two beta chains, pictured in green, so that its overall composition is referred to as alpha 2 beta 2. In a previous lesson, we reviewed the structure of myoglobin, another oxygen-binding protein. Recall that myoglobin is a monomer and would therefore not exhibit the quaternary level of structure. Hemoglobin is also an oxygen-binding protein, and it's interesting to note that the two chains in hemoglobin and the one chain of myoglobin are very homologous, that is, similar in three-dimensional structure. With its single heme group, myoglobin can only bind one oxygen molecule at a time. In contrast, hemoglobin carries four heme groups, one in each subunit, and carries four molecules of oxygen when fully saturated. This strikes us immediately as being optimized for oxygen pickup and delivery, which is indeed the function of this very interesting protein. In our next lesson, we'll see the effect of these structural differences of myoglobin and hemoglobin on their respective functions.